Next presenter is Julia Omotade.
Thank you everyone in advance for your attention. My name is Julia Omotade. I'm a third-year graduate student from Emory University in Atlanta, Georgia. And today I will be speaking to you about the role of tryptophan, which is an actin binding protein, in the establishment of synapses within the mammalian brain. And before I begin, I would of course like to thank the organizers of this conference for the opportunity to present my work here with you all. So the human brain is an extraordinarily complex organ that's comprised of over 100 billion neurons. And neurons such as these form extensive neuronal networks, and it's critical that these neurons be able to communicate in a precise, coordinated, and rapid manner. And this task of neuronal communication is largely accomplished through the chemical synapse. The chemical synapse is so named because chemicals in the form of neurotransmitters are released from the presynaptic axon terminal. The dendrite must be able to receive and respond to this input, and it does so through these tiny structures covering the surface of the dendrite referred to as dendritic spines. Although small, a close-up reveals that dendritic spines actually have all of the biochemical machinery that's needed to respond to this input. So on the plasma membrane, you have neurotransmitter receptors that can actually bind to the neurotransmitters. And there's actually a very dense array of scaffolding proteins, signaling proteins, and other proteins right underneath the membrane that's able to transmit the signal. So the dendritic spine thus represents the postsynaptic component of most excitatory synapses. And in this way, the brain is able to modulate the synapse on a spine-by-spine manner. So as you can imagine, the establishment and connectivity of these synapses are very tightly regulated. And what happens when you have synapse dysregulation, such as synapse loss or aberrant synaptic signaling, is that you get a host of neurological diseases. So something that you may have noticed in the previous picture is that the dendritic spine has a very stereotypical structure. And something we've known for actually over a century now is that this structure is critical for spine function. And what you see is that in a variety of neurological diseases, you have changes in spine density and spine shape. And so we've known for a long time now that spine structure is tightly coupled to synaptic activity. So then the question becomes, what regulates spine structure? And the answer is the actin cytoskeleton. So the actin cytoskeleton is the most abundant cytoskeleton element that's found within dendritic spine. So it's highly concentrated within the structure. And within the neck here, it's organized into linear filaments, and within the head, it's organized into a more cross-linked network. And so there's a very important aspect of synapse biology by which scientists are trying to figure out the mechanisms that regulate the actin cytoskeleton within spines, seeing as actin gives the spine not only the structure, but it helps regulate the organization of receptors, as well as other proteins within the postsynaptic compartment. And so that brings me to the focus of my talk. So my lab is interested in understanding how the actin cytoskeleton is regulated during spine development. So dendritic spines here aren't simply just born or emerge from the dendrite. Rather, they undergo a very stereotypical pattern of development. So in early development here, highly motile and dynamic filopodia arise from the dendrite. And upon contacting the axon terminal, represented here, they undergo distinct morphological events that leads you to the mature spine. So this includes head shortening and also extension. And so what you can think of is that the conversion from a very motile filopodia, seen here, to these stable spines that you have in the adult brain represents a stabilization event by which you have the dynamic actin skeleton that needs to be stabilized by a host of actin binding proteins. And so here I'm showing you an EM image of the actin cytoskeleton in filopodia, and you can see that as opposed to the linear filaments that you see in filopodia, in the mature spine you have a predominantly branched network. And so, as you can imagine, there's spatiotemporal regulation that activates binding proteins to spur this change. And so my lab is focused on the actin regulatory mechanisms that happen during dendritic spine development and synapse formation. But before I launch into my data, I need to acquaint us all with some basic actin biology. So within the cell, actin is present in two forms. You have G actin, globular actin, which polymerizes into filamentous actin. And something that's a critical aspect of the actin filament is that it's polarized. And so monomers are added to the plus end, or barb end, and subunits are taken away from the minus end, so that is a site of depolymerization. And so within the cell, the actin cytoskeleton is not static. It's actually rather dynamic, and this dynamic nature is imparted by actin-binding proteins. And a very particular class of actin-binding proteins are called actin-capping proteins. And so what actin-capping proteins do, they either bind to the plus end or the minus end. And proteins that bind to the plus end actually stop polymerization, thereby restricting it to where it's needed. And so you have spatial and temporal control in the cell. And equally important, you have proteins that bind to the minus end. So this is going to inhibit depolymerization, stabilizing filaments, and promoting growth. And so even though there's a host of proteins that have been shown to bind to the plus end, there's only one known molecule that binds to the minus end, and this is called tropomodulin. And this has been shown to be critical in a variety of cell types. So that brings me to a more specific goal, to understand the role of tropomodulin, this minus end cap, during dendritic spine development and synapse formation. And so there's 4 isoforms of tropomodulin, but I'll be speaking about tropomodulin 1 and 2, which are the neuronal isoforms. And so, very briefly, this is a schematic of the domain structure. And the only thing I really want to draw your attention to here, although it has a lot of domains, is this actin-binding domain. And so this is important for it to physically bind to the pointed end and impart control on the filaments. Okay, so how might tropomodulin actually regulate actin dynamics? So here in this scenario, we have a filament that's completely uncapped. So as I told you, the plus end is going to be the site of polymerization, whereas subunits will be lost from the minus end. However, if you add tropomodulin, you can have a scenario where A, you cap the minus end, and then you favor net polymerization of filaments, or in the case where the plus end is also capped, you'll have no monomer exchange at either end, and this will lead to very stable filaments. So in this way, tropomodulins are able to promote polymerization and stabilize filaments in a wide variety of cell types. So even though we know this molecule is important, intriguingly, we know almost nothing about what it's doing in the brain. So we know that it's highly expressed in CNS, it is there. We know that a tropomodulin 2 knockout mouse has defects in learning, memory, and synaptic plasticity, so we know it's important. We also know that the protein levels are misregulated in various diseases, but we still don't really know what it's doing, what are the subcellular localizations, and how it functions to regulate the actin cytoskeleton. And so we still have many outstanding questions. Is tropomodulin expressed in dendritic spines? At what time is it expressed? And how does it regulate the actin cytoskeleton? And so the first question, is tropomodulin even expressed in dendritic spines? Really briefly, I want to acquaint you with my model system. So for the remainder of my talk, I'm going to be showing you primary rat hippocampal neurons. And so how I get this is I dissect out the brain from an embryonic day 18 rat. I further take out the hippocampus, and I plate these on cover slips. And as you can see here, these neurons are able to grow on these cover slips and form very mature dendritic arbors, which actually have dendritic spines that occur at the same density and same size that are seen in vivo. So this culture system provides an excellent model for me to study the development and also mature spines. And I'll be using images such as these in the remainder of my talk. Okay. So what did we find? So of the 4 isoforms, we found that tropomodulins 1 and 2 are present within mature dendritic spines. And so here in the first panel, I'm showing you dendritic spines, which are marked with phalloidin. So this stain selectively binds to actin, and so it's used to demarcate the dendritic spine. And as you can clearly see, tropomodulin 1 here localizes very nicely within dendritic spines. And it also localizes very nicely with PSD95, which is another marker of dendritic spines. And for the sake of time, I will be either selecting images of tropomodulin 1 or 2, but they both have very similar distributions within dendritic spines. Okay, so because I do a lot of confocal microscopy, actually the last slide I showed you was confocal microscopy, I have the advantage of being able to render 3-dimensional images. And so this makes it easier to see the actual localization of this protein within dendritic spines. So here in green is my molecule, tropomodulin 1, and here in red we have the marker for the dendritic spines. And even though we found that tropomodulin is in spines, we found something that was rather intriguing. So instead of the protein just being in the entire spine, as most proteins are, and most proteins are either in spines or they're not in spines, within the spine itself, we found a very intriguing distribution of this protein. So if you would please play the video, I can demonstrate that. So it's the slide before, so yeah. The video is also on the desktop. I've minimized it, so perhaps we can get it that way. Thank you for your attention. Maybe we'll just skip ahead to this slide. I think I can still demonstrate it with this thinking of. Okay, so we found something that was really intriguing, actually. So typically within spines, proteins are either there, as I said, or they're not. But what we found is that tropomyosin actually selectively localizes to the core and the base of the spine. However, it's excluded from the outermost tip, as you can see by the failure to colocalize with the red signal, which marks the entire dendritic spine. And so why might this be? Well, we think perhaps tropomyosin is associated with two distinct pools of F-actin within the spine. So within the dendritic spine, although I said that actin is extraordinarily abundant, the actin there is not simply a homogeneous population. You have a stable pool towards the core, and you have a very dynamic pool towards the top of the head. And so perhaps tropomyosin is localized to the core because it's helping this pool of actin to be more stable by stopping depolarization, whereas it's absent from the dynamic part of the spine because obviously if those are rapidly treadmilling, you would not want to cap the point end, you would actually want to promote the depolarization. So we think that this is really exciting. Not only is pointed end capping a mechanism used in synapses to regulate the actin cytoskeleton, but perhaps there's two different functional pools that relies on this molecule in a different way. And so, but as I told you, I'm interested in development, and so we made a very simple prediction. If tropomyosin is participating in development, it should be expressed during development. And so for this, we did a Western blot of brachiohippocampal lysates, and so these stages correspond to before, ongoing, and complete synaptogenesis. And so as you can see, during these time periods, tropomyosin 1 and 2 are both expressed, but tropomyosin 1 actually being developmentally regulated. And interestingly, the expression of tropomyosin peaks during the time of synapse refinement and extensive synapse formation. So perhaps that's the molecule that's more specific in synapse formation as opposed to just maintenance in adult cells. Okay. But obviously, I need functional data, and so I was able to generate knockdown constructs for tropomyosin 1 and 2. So after a lot of trial and error, I was able to create a hairpin 1366 that's specific for tropomyosin 1, but does not knock down tropomyosin 2. I was also able to generate a hairpin 661 that is specific for knocking down tropomyosin 2, but does not knock down tropomyosin 1. And with this, what I plan on doing is introducing these constructs into developing neurons, so DIV14, that would be days in vitro 14. And so when I introduce this tropomyosin shRNA, I'm going to be looking at the effect on the structure of dendritic spines. So I hypothesized that without tropomyosin, it would lead to uncapped filaments and thus increased depolymerization, thereby affecting spine structure. Also affecting the organization of the postsynaptic density components shown here in receptors and molecules, and also ultimately affecting synaptic transmission, which is the functional output. So in conclusion, we've shown for the first time that tropomyosin 1 and 2 are indeed localized to dendritic spines, thus pointed end capping could be a mechanism by which the actin cytoskeleton is regulated within the synapse. Tropomyosin have a distinct distribution within the postsynaptic compartment, perhaps associated with two different kinetic pools. And in the future, of course, I will use my knockdown constructs to actually examine synaptic function and morphology. I will also be overexpressing my GFP tag tropomyosins, which I was just able to generate for tropomyosin 1 in order to do the corollary experiment to the knockdown. And then, of course, I will be doing live cell imaging using these tropomyosins to see where in development tropomyosin molecules enter the spine and how they're regulated in accordance with actin distribution. And with that, I would like to thank my lab, my collaborators, my inviting me to speak about my research, you all for your attention and my funding. And I'll take any questions.
Could you tell us, I have two questions. Could you tell us a little bit more about the T-mod null mice? Do they have some kind of physiology or what are their spines formed? And also, have you considered using just some kind of pre-loss model versus construct-based construction T-mod?
Okay, yeah. So the tropomodulin 2 null mice has no gross anatomical abnormalities. But there is hyperactivity, there is learning and memory, there are learning and memory challenges. And there are also, learning, memory, and hyperactivity. In regards to the dendritic spines, no one has examined the synapse, no one has done EM studies, no one has looked at that at all. So although we know that there's functional outputs, no one has looked at the molecular mechanisms, and certainly not dendritic spines. And then the second question, pre-loss. So no, we're more of a cell culture model because we actually want to delve into the molecular mechanisms. What is tropomodulin associating with? What are the molecules at what time point in development? And so for that, using an shRNA system is actually the best model for us. And it also allows us the flexibility to transfect at different stages and to track what tropomodulin is doing during the entire process of development. Jamie?
So you mentioned you plan on using your GFP tag tropomodulins for the lab. So do you plan on looking at any other cytoskeletal proteins?
Yes. So when I transfect the GFP tag tropomodulins, I'll also transfect the LIFACT, which is an F-actin marker, so I'll be able to demarcate in real time the actin cytoskeleton, and I'll be able to compare tropomodulin in relation to changes occurring in the root stomach. Any more questions? Brenda?
Is there any knockout for tropomodulin in life?
No, so there's no knockout for tropomodulin 1. They chose the tropomodulin 2 to be knockout because that's a strictly neuronal isoform, whereas tropomodulin 1 is also found in other generally differentiated post-narcotic cells. But something interesting though, there was an 8-fold upregulation of tropomodulin 1 and the tropomodulin 2 null mice, which really calls for a culture-based system to tease apart the isoform-specific effects, which is again why we're using cell culture. Move on.
Julia, what are your presentation in your cell culture model? How do you determine when development starts? Because you're taking these out of E18 rats, and then so your phase of future 14. How is development determined?
Yeah, so that's a great question. So during the E18 rat, essentially synaptogenesis has not started at all. So you get to track all the process starting from axon cap binding up until the establishment of synapses, to the stabilization of synapses, and then mature synapses. So we're actually seeing it from the very beginning, starting from the growth cone up until mature spine. So in that very short amount of time, of 21 days, you can mimic like the embryonic postnatal and what you would see in the actual adult rat. Okay, thank you very much.